Here is the question. Question is column matching. I have already written the question. The question is for Neet September 2020 exam questions. I'm discussing it. Okay. Here, in the column 1, different bacteria have given, like Bacillus thuringiensis, Thermus aquaticus, Agrobacterium tumefaciens, and option D has given Salmonella typhimurium. And column 2 has given cloning vector, construction of fast DNA polymerase, cryprotein. Column 1 has given the name of bacteria which we have to match with column 2 by their biological use. Okay, that is in which purpose these bacteria are used. Okay, first, Bacillus thuringiensis. You know that Bacillus thuringiensis bacteria contain a protein which act as insecticide or insect resistance, that is called as cryprotein. Here I am matching it for you. Yeah, this. Okay, now come to Thermus aquaticus. This is also called as Taq polymerase. Okay, it has the capacity to resist high temperature. That's why it is used in DNA polymerase or PCR process. Okay, the primer used which should be thermostable. So that PCR, that is polymerase chain reaction purpose, Thermus aquaticus has used. Okay, this is DNA polymerase. Now come to Agrobacterium tumefaciens. Agrobacterium tumefaciens is a cloning vector. Okay, it is found, used in clone in plant DNA. Okay, here, and Salmonella typhimurium. Salmonella typhimurium is used for fast DNA construction. Okay, this is option 2. I have already matched it. I'm explaining all the point here. Okay, first Bacillus thuringiensis, which I have here. You can see it. Bacillus thuringiensis, which I have matched with IV point of column 2, that is cryprotein. Here I am writing it. Answer. Cryprotein is a toxic crystal protein. Okay, it is encoded by crygene. Encoded by crygene. The crygene is insect resistant. Okay, insect resistant but harmless to the plants. Okay, how it becomes work? The cryprotein is produced by the protein is produced by Bacillus thuringiensis bacteria. Okay, Bacillus thuringiensis bacteria. But it is in the inactive condition. Okay, inactive condition. When it is ingested by insect, the bacteria bacterial protein, okay, that is cry protein, gets activated by the low pH of gut of insect, pH of insect gut. Thus, it becomes toxic to the insect and kills it. While the cry protein remain harmless to the plant, okay, in which it is injected. Thus, it acts as an insecticide. Now, let's come to the second one, that is Thermus aquaticus. Okay, let's discuss it. Okay, Thermus aquaticus, we found this bacteria in hot spring, and they has the capacity to resist high heat. Okay, and for this purpose, it is used in PCR process. Thermus aquaticus is a bacteria found in hot springs. Hot springs. It can resist high heat, due to presence of enzyme Taq polymerase. Okay. This is Taq polymerase. Hence, Taq polymerase is used in PCR. Okay, this is the reason. And now come to the next bacterium. Here you can see next bacterium is Agrobacterium tumefacium. Okay, let's move to that one. Yeah, Agrobacterium tumefacium is a soil bacterium and used as cloning vector. Okay, cloning vector. It is used to transfer a small segment of DNA into plant genome. By the process known as transformation. Okay, this is Agrobacterium triumphacum is used as the vector, cloning vector. Here I'm matching it. This Agrobacterium triumphacum cloning vector. Now last one, Salmonella typhimurium. Yeah. The fast recombinant DNA was constructed by Stanley Cohen and Hubbard Boyer in 1972. Okay, they cut the piece of DNA from plasmid carried antibiotic resistant gene. Okay, the fast recombinant DNA was constructed by Stanley Cohen and Herbert Boyer in 1972. They cut piece of DNA from plasmid. You know that plasmid has extra chromosomal DNA is present in the bacteria. Okay, and it carries antibiotic resistance gene. And where this gene is found? In the bacterium Salmonella typhimurium. And linked it to the plasmid of E. coli. Okay. This is all about the explanation of this column matching. You can see it here. And this is the column which I had already matched. And these are the explanation. Okay. I hope it is clear now. This is the last explanation. Yeah, let's see. Okay. I hope it is clear now. Thank you for watching. Bye.